Hello, my name is Javier Perez Florido from the Clinical Bioinformatics area, Joaquín Dopazos Lab, and I'm going to present the work entitled CSVS, the Spanish Population Variability Database and the First Spanish Genome Reference Imputation Panel. So, advances in the field of genomics over the last 25 years have led to substantial reductions in the cost of genome sequencing. It is well known that sequencing technologies have experienced an unprecedented development during the last 10 years that resulted in different international collaborative projects which contributed to an extraordinary increase in the knowledge of the mutational spectrum of diseases. A couple of examples of these collaborative projects are the 1000 Genomes and GenomeAD projects, among others. This generation of knowledge has been especially significant in diseases with high morbidity and mortality caused by highly penetrant variants. In fact, more than 4,500 monogenic diseases can nowadays be directly diagnosed by personalized genomics. Among the strategies used to discover new disease variants, especially in monogenic disorders, frequency-based filtering has demonstrated to be a very useful tool. Why? Because variants that are relatively common in a control population are likely benign, while rare variants, especially if they have functional consequences found in multiple affected cases but absent in the control population, are likely to cause disease. Therefore, it seems clear that the availability of healthy controls is a decisive factor for the discovery of new disease determinants. From an historical perspective, the 1000 Genomes project produced the first comprehensive catalog of common human genetic variation. However, it is known that low frequency and rare variants, typically population-specific, are poorly represented in such catalog. Actually, recent studies have described a remarkable local component and a high stratification level in many rare variants with uncertain functional consequences. As a consequence of this, the risk of many diseases differs in distinct human population according to their genomic genetic backgrounds. In fact, the knowledge of the genetic variability of the local population has been revealed as a critical factor for the discovery of new disease variants. All these observations highlight the need for population-specific catalogs of genetic variation. However, only a few initiatives to study genetic variation at the population level have been carried out today, which include, for example, the genome of the Netherlands, the Medical Genome Project here in Spain, the 3000 Finnish genomes, or the Icelandic population study, among others. In spite of its recognized usefulness, large-scale sequencing projects of cohorts of local healthy population require expensive consortium-based projects to obtain a representative sample of the population targeted. Unfortunately, funding bodies tend to be reluctant to fund projects that involve systematic sequencing of healthy individuals. In this scenario, a crowdsourcing strategy can provide a feasible alternative to traditional working schemas by organizing consortia that collect data from different groups that are collectively benefited of the sample size. And this is the case we present here, a crowdsourcing database of the Spanish population genetic variability, which is called the Collaborative Spanish Variant Server. Currently, CSVS stores information of about 2,000 genomic and genomic sequences of unrelated Spanish individuals contributed by different consortiums and projects, such as the Medical Genome Project, the Undiagnosed Rare Disease Program from Ciberer, the Project Genome 1000 Navarra, and Rare Genomics, among other research groups and initiatives across Spain. The repository, which contains detailed allelic frequencies corresponding to the aforementioned sequences is used as a pseudo-control population for finding new disease-causing variants and genes, with this idea: disease A is a healthy control for disease B. Despite gene pleiotropy cannot be completely ruled out, data are being at higher disease ICD chain categories. This way, aggregated counts of allele frequencies of the pseudo-control Spanish population can be obtained for diseases belonging to the category removed. This is how CSVS looks like. The initial screen requires the acceptance of the terms and conditions for the use of the database. Before starting any operation, once accepted, different options can be used. The search option here allows querying the CSV database. In the left panel, queries can be done by gene symbol or by chromosomal regions. Also, one or several disease categories can be excluded, and variants can be highlighted using different types of score, CIF, polyphane, CAT, etc., as well as sequence ontology terms for the variation consequences. For example, we can select variants for a given gene or chromosomal region and remove a specific ICD chain category of interest. In this way, aggregated counts of allele frequencies of the pseudo-control Spanish population can be obtained for diseases belonging to the category removed. The results of the query include a list of positions for which variation has been found in the Spanish pseudo population, along with complementary data such as chromosome, position, reference allele and alternative allele, allelic frequencies in the Spanish population, allelic frequencies in the 1000 genomes population and in the exact and ESP population, impact and conservation indexes, CIF, polyphane, CAP, etc., the consequence types assigned to the mutation, and the phenotypes corresponding to known clinical information for the variants extracted from ClinVar, Cosmic, and all this information are annotated interactively on each query using the cell-based web services. Also, a visualization of the variant in the genomic context is provided based on the Genome Maps browser. Additionally, some extra detailed information can be found on the population, frequencies observed for the variant, the phenotype, or the effect. An interesting option is the contact request button, offered for any variant in the query results panel, which is a local equivalent of a matchmaker exchange service, extensively used to contact the original contributor of a specific sequence. The stats option provides an updated view of the content of the CSVS database. For example, the distribution of variants per ICD term in CSVs, or alternatively, the distribution of samples per ICD term. Saturation plots provide an interesting perspective on the general conservation of the gene studied and consequently on the possibilities of discovering new variants into it. Genes highly constrained to change will saturate soon, and a relatively low number of individuals will capture most of the tolerated mutation the gene can handle, while unconstrained genes will present a still growing slope, meaning that there are still many variants that can potentially be discovered. Discovering a new variant in a saturated gene can be more relevant than the same finding in a non-saturated gene. This is an example on BRCA1 gene, which seems quite saturated. Partial or total downloads of the aggregated data are possible upon the reception of the corresponding data download agreement DALI site. So you can use this data for your research. How CSVS is populated with data? Briefly, this is the data workflow. Here, these are the different genomic projects that provide data to CSVS, and some QC is run before variants are aggregated to the database. The first QC procedure includes an artifact and kinship detection test. This test determines undesired samples based on the percentage of novel variants introduced in the database, either by excess, which could be a potential noisy sample, or by defect, corresponding to close relative or individual already in the database. The second QC procedure consists of testing the Spanish locality of the samples. For this test, sequences belonging to different populations in the 1000 Genomes project were used to train a machine learning-based decision model to discriminate Spanish samples from the rest of populations. Briefly, the individual ancestry in 1000 genomes data was estimated for 26 subpopulations using admixture. The goal was to learn population structure from the 1000 Genomes Reference Panel and project the CSVS individuals on it. Finally, if the VCF file passed both tests, the variants are stored in the CSVS database and allelic frequencies are updated according to these variants. The genomic data from CSVS has been used to define the first Spanish Genome Reference Panel for imputation. Genotype imputation is an essential component of genetic association studies. This way, two alternative reference panels using the Minimac3 imputation tool have been created for comparison purposes that include CSVS whole genome sequence variant panel composed of 228 samples plus either the entire 1000 reference panel or exclusively the Spanish population contained in 1000 project named as IBS. The original 1000 genome panel was also used for the comparison. So these panels were compared in terms of imputation accuracy, which is the correlation between real and imputed genotypes, known as genotype correlation. For this comparison, 32 samples were extracted from the set of samples, and the reference panels were built with the remaining ones. All the positions from the 32 genomes samples were imputed. We saw here the results on the 4 longest chromosomes. Variants, SNP and indels were gathered by their minor allele frequencies. As you can see here, the imputation improvement is even more significant when variants in rare sites were included, increasing the accuracy from 65.8% to almost 90% in the 4 chromosomes. Also, there were no significant differences in terms of imputation accuracy between both Spanish reference. It must also be emphasized that another reference panel was recently created using nearly 1,000 exomes from CSVS. Finally, I'd like to emphasize that CSVS makes its genomic information discoverable through the GI4GH beacon network. Either you can search variants in CSVS through the beacon network website, together with other beacons, or using the available API on the second link. In either case, a yes, presence of variant, or no, absence of variant response is provided. To sum up, the genetic variability of the local population is recognized as one of the most relevant factors in the discovery of new disease variants, especially in Mendelian diseases. Genomic data of healthy individuals belonging to the local population of interest are often scarce when not unavailable. The CSVS provides an original solution to this problem. The database is a continually growing resource that collects genomic or exomic sequences of the Spanish local population, no matter whether these come from healthy or diseased individuals. And it is an example for the construction of population-specific pseudo-control repositories by means of crowdsourcing. Finally, this work has recently been accepted in nucleic acid research and has been published a couple of days ago, and you'll find more results in the main paper. So thank you for your attention, and now it's time for questions if you have some.